Hi, I'm Tony, and I'm going to be discussing the thyroid today. We're going to be paying particular attention to the function of the thyroid, the hormones it releases, and how these hormones are essentially synthesized. At a later lecture, I'll be discussing how problems with the thyroid can manifest to different disease states and treatment options. But for now, let's get started. So the thyroid is an endocrine gland, and an endocrine gland is one that simply secretes its products, in this case hormones, into the bloodstream directly. The thyroid regulates the metabolic rate and synthesis of proteins using the hormones it produces. These hormones begin production in the follicular cells, which are a type of cuboidal epithelium, and the whole system is governed by a negative feedback loop involving the hypothalamus, pituitary, and thyroid. In the anatomical picture here, you can see that the position of the thyroid sits directly below the Adam's apple and sits on top of the trachea, which is of course the main breathing tube in humans. So the follicular cells in the thyroid produce two main hormones, and they're known as triiodothyronine, T3, and thyroxine, T4. And the main way we can distinguish between these two is the amount of iodine found attached to the core benzene structure of each of the hormones. So on the left hand side we have T3, and I've just circled the iodines that are found in T3, and as you can see there are three of them. That's why it's known as T3, triiodo T3. And on the right hand side we have thyroxine, known as T4. And again, I've circled the iodines found attached to the core benzene structure, and they are essentially four of them. So T3, T4, the number essentially refers to the amount of iodines found, and that's how we can distinguish between the two different types of thyroid hormones that are produced by these follicular cells. To begin the production of T3 and T4, you first need iodine to be taken up by the follicular cells, which is a critical component of these hormones, as what we just saw in the previous slide. These follicular cells are arranged in a simple halo ring structure where the space inside the ring is known as a thyroid colloid. Think of it as like an empty space. And this is where the production of T3 and T4 occurs in the later phases. So here on the right you can see the bloodstream, and iodine which comes from our diet circulates here. When it comes to a follicular cell, it crosses into it via the iodine-sodium symporter, which transports one iodide and one sodium into the follicular cell. So simultaneously as iodide and sodium enter the follicular cell, the protein thyroglobulin is being transcribed and translated. The nucleus of the follicular cell begins this process, and the thyroglobulin is stored in vesicles. So to understand why thyroglobulin is important, we have to look at its core structure, or zoom in on the structure. Now the thyroglobulin protein contains amino acids, and one of the amino acids of focus is tyrosine. So as we can see in this picture here, the tyrosine residuals form the core of T3 and T4, and the benzene ring structures shown in these tyrosine residuals are where the iodine will eventually bond to form the precursors to T3 and T4, which we'll look at a later step. So upon transcription and translation of thyroglobulin and the uptake of iodide, both make their way from the follicular cell into the thyroid colloid via exocytosis and the iodine channel transporter respectively, as seen here in the animation. So as iodide comes through the channel, it needs to be made more reactive, and we want it to be made more reactive because we need it to bind with thyroglobulin. The enzyme that is responsible for this process of making it more reactive is thyroid peroxidase. In the presence of hydrogen peroxide, thyroid peroxidase oxidizes the iodide to become iodine. What this means is an electron is lost and the iodide ion becomes the element iodine. Simply, when we take away one electron, we add a +1 charge, and this makes it neutral in the case of iodide. And because there is no charge, the iodine is in its elemental form. The elemental form of iodine is highly reactive, and the thyroid uses it to its advantage in the production of T3 and T4. So the highly reactive iodine goes fishing for something to bind to, and the primary target, which we're interested in the formation of T3 and T4, are the tyrosine residuals which form part of the thyroglobulin. Now here in this slide, we've just focused on tyrosine here. There's obviously other amino acids that form thyroglobulin, but the tyrosine amino acid is what we're interested in because that is the precursor to T3 and T4. So depending on the amount of iodine bound to the benzene ring core of tyrosine, we give it a certain name. So if there's one iodine bound, we call it monoiodotyrosine, MIT, and if there are two iodine bound, we call it diiodotyrosine. And as you can see, these are starting to be very similar to the structures of T3 and T4, but we're not quite there yet. We're almost there, but not essentially there. So here we have our thyroglobulin, and the tyrosine residuals have reacted with iodine to form varying amounts of MIT and DIT. Now, the last step occurs in the thyroid colloid is conjugation. So here, MIT and DIT come together to form structures very similarly to T3 and T4. The only difference being is that they're bound to the thyroid globulin. This occurs somewhat randomly. So you can have either an MIT bind with a DIT or conjugate with a DIT to form pre-T3. And as you can see, 2 iodines plus 1 iodine is equals to 3 iodines. So that's how you have your pre-T3. Or you can have 2 DITs come together to form pre-T4. And this, as you can see, is your 4 iodines. Now, as you can see, these are essentially T3 and T4. The only difference being is they're bound to the thyroid globulin. So obviously there's going to be a step that occurs where the thyroid globulin is separated from these tyrosine residuals which have reacted with iodine and have come together to form pre-T3 and T4. So the thyroid globulin containing the pre-T3 and T4 enters back into the follicular cell via endocytosis, where one of the final stages begins. So here we need to separate the T3 and T4 from the rest of the thyroid globulin components, and this occurs via proteolysis, which, as its name suggests, lyses proteins or cuts specific components of the protein to give what we want. This allows for T3 and T4 to be separated from the remaining thyroid globulin, whose components can then be recycled by the cell for the next line of production of further T3 and T4. So the final step is that the T3 and T4 hormones make their way out of the cell via monocarboxylase transporter, where they'll eventually exert their mechanism of action and mediate effects on the rest of the body. So now we know how T3 and T4 are made by the thyroid, we can look at some of the differences between the two hormones. Structure-wise, we kind of already know the difference, we've gone through it already. Essentially, you want to look at the amount of iodines bound to the benzene ring structures that form the hormone. So in T3, 3 for 3 iodine, T4, 4 for 4 iodine. Potency and amount produced by the thyroid vary quite a bit between the two. Now if we look at the potency, we're looking relative to T4, so we assume T4 is a potency of 1. Triiodothyronine, or T3, is approximately 3.3 times more potent than T4. And inversely, we're going to have less produced by the thyroid. So the amount produced by the thyroid differs. So T3 is about 20% of the T3 and T4 is T3, and 80% of the T3 T4 is actually T4. So they're the main differences between the two hormones produced by the thyroid. So we saw how T3 and T4 are excreted from the follicular cell. Now we need to look at how they are transported in the blood, how they bind to their cell targets, and the effect they exert. Due to their lipophilicity, and this comes from parts of the structure of T3 and T4 like the benzene ring, T3 and T4 need to be bound to particular circulating components of the blood and do not get transported unbound. These circulating products include albumin and thyroid binding protein. Once they reach their cell target, they enter the cell via a family of cell transporters known as the monocarboxylase family. And this is similar to the transporters found in follicular cells, which transport T3 and T4 out of the cell. When they enter the cell, they bind to nuclear receptors, known as thyroid receptors. The T3 or T4 thyroid receptor complex initiates gene promoters, and this cascades to the enabling of transcription and the translation of proteins. So T3 and T4 increase the basal metabolic rate, and this increases the body's oxygen and energy consumption. So if you think about it, people who overexpress T3 and T4 are going to lose a lot of weight in a short period of time, because the basal metabolic rate is the minimum amount of calories to sustain someone at rest. When you increase that, you're burning more calories when you're at rest, and this essentially means you're going to lose more weight in a shorter period of time. So what T3 and T4 also do is they increase cardiac output. This can increase the heart rate, and this can give you things like high blood pressure, tremors, and that sort of effect on the body. We also see the increase in the metabolism of glucose, which again can be attributed to weight loss. And further, it stimulates the production of RNA polymerase 1 and 2, and this increases protein synthesis, so it creates a host of other proteins with other functions in the body. And again, when you disrupt the thyroid, when you disrupt the negative feedback mechanism between the hypothalamus pituitary and the thyroid, you're going to have side effects which are attributed to this, what we see in this slide here. I'd like to thank you for listening. I apologize. I'm not the most confident when talking, but hopefully I'll get better with time. I'll be making another lecture on the different diseases that can occur, that can manifest as a result of thyroid problems. And yeah, catch you later. Have a good one.